one has to worry too much about how we define it. Clearly, biology has changed over the years. There are things that we have to do using computational approaches and algorithmic approaches. Slowly over time, I think all biologists are going to rely more and more on computers and computational analysis than just test tubes and petri dishes. So in some ways, it's a natural movement of phenomena of biology reflecting really that cells, human cells, eukaryotic cells, are some of the most complex things in the known universe. And really, to be understanding them and analyzing them, we're going to have to draw on a lot of computational approaches and computational modeling, and storing our data in large disk arrays and using disk farms and compute farms to actually analyze the data that we're generating. Some eminent bioinformaticians have said that maybe the term would just go away, and really it's just another way of saying biology. My interest was really piqued by the idea of DNA as just storing information. And we were getting to grips with the kind of the sizes, the amount of information that was in the genomes. And I was just so intrigued by what is in these genomes. And wanted to study the genome just for the genome's sake, just to find out what information was there. And so that's what made me jump from biochemistry into genetics. And I went to Simon Fraser University to study genetics. And there's a laboratory there, Professor David Bailey, who was doing something I really wanted to do, which was just look at the genome for the genome's sake, to look at what is the information that was there. Around that time, the genome project, the Human Genome Project, was starting, but it was embarking on model organisms first, much more tractable, smaller genomes. And so C. elegans, which was the organism I worked on, was one of those chosen to be sequenced. That's essentially why I ended up doing bioinformatics, and it wasn't really a bioinformatics. Bioinformatics as a field didn't really exist when I started to do that. Well, the big projects currently are really that we can now go into human cancers and characterize all the genetic changes that have accrued within those cells during the onset of the disease. So really this is opening up the whole disease to a level of interrogation that we've never had before. And that's giving us huge insights into ways in which we can come up with better prognostics for disease, better diagnostics, and certainly, hopefully as well, better therapeutics. At this point, we've just started with our first tumors. We've got tumors probably in the tens now from different sites. And I think as we build up to getting maybe samples or completed genomes of, say, 100 different breast cancers or 100 different thyroid cancers, that's going to give us some real powerful statements to say about what kind of molecular changes the patients have. And should those changes correlate with different therapeutics or different strategies? Can we make sure that every patient gets the personalized approach to curing their tumor that the molecular biology suggests? Cancer is obviously a huge healthcare issue, and obviously hits many people at a very personal level. There's huge impetus to apply bioinformatics tools, DNA sequencing technologies, to really trying to understand the molecular mechanisms within cancer. So with that impetus, what other things do we can learn? I guess another question is what could be the benefits from that effort? We're studying cancer at the DNA level, but all organisms have DNA, and so we have lots of examples where some of the pipelines we've created to study human cells, we've been able to apply in other fields, in forestry and fisheries. Some of the assembly algorithms that we were developing here to look at what we call translocation breakpoints in human tumors we were actually successfully able to use to assemble the fungus which the mountain pine beetle actually injects into the trees as they move from tree to tree, and it's the fungus that contributes part of the pathogenicity or the death of the tree. The fact that we're dealing with DNA means that we can transfer these skills actually quite easily from different fields. Just realize, that where biology is taking us and bioinformatics, and the potential is huge in terms of healthcare now that we have the technology to sequence complete genomes, and we have a complete human reference genome, for instance, that we can base all our studies on. There's just a huge amount of analysis to be done, either in the wet lab or computationally.